Hello everyone, my name is Dr. Yuxiao Chen, and I am a postdoctoral research fellow in the lab of Dr. Tripathi. Today I will be sharing with you some of the work that I have done as a CONP scholar. In particular, I will be talking about my work on improving the quality, documentation, and dissemination of multimodal single-cell data. So multimodal data or data sets are basically data sets that contain information across a variety of experimental setups or different types of information. And these kind of data sets are more and more common as researchers tackle research questions with more cross-disciplinary approaches. The specific type of multimodal data that I am going to be talking about today come from patch-seq experiments. That is shown here in this diagram below on the bottom half of this slide. And what patch-seq experiments are basically building up on top of traditional patch clamp electrophysiology, where neurons are patched and their electrophysiological properties are taken and measured, where we then, on top of that, take the individual cell and sample their RNA. And more specifically, we take these samples and we put them through single-cell RNA-seq. So patch-seq is particularly important because it allows us to connect the expression of genes from DNA with the actual basic functions of neurons within individual cells. However, no current consensus gold standard protocols exist for the data processing or data quality control from patch-seq experiments. Previously, Dr. Tripathi from our lab, the PI of our lab, pioneered a protocol for quality control of such patch-seq data, and my work that I'll be sharing today is basically about further development of this data processing and quality assessment pipeline. Now, we are particularly focusing on the RNA-seq aspect of this data set, particularly because single-cell RNA-seq data is very sparse and sensitive to technological variation. As part of this process, we are documenting our pipeline development with annotations for improved generalizability so that other labs can follow in our footsteps or can use our pipeline or can generally understand our rationale and decisions that we made in the overall process. And finally, we aim to disseminate our data and pipeline in an open and accessible manner so that the broader research community can really quality control and process patch-seq data or really RNA-seq data in general in a much more efficient and effective and reproducible manner. So our methods involve identifying, assessing, and benchmarking tools for RNA-seq data processing and quality assessment. We will be testing these tools with both novel and published RNA-seq and patch-seq datasets. And again, we will be compiling all of our results and data and decisions and rationale and so on and so forth in the form of documentation and recommendations that we will be sharing publicly. So this is an example of an output from our pipeline. And this is a simple and intuitive example. What we see here is data collected from a pilot patch-seq experiment from our collaborators in the lab of Dr. Simmons from the University of Heidelberg. What we see here are patched samples as well as negative controls. And what we basically see is a breakdown of how all of the RNA-seq fragments from their patch-seq experiments align when we try to align them to our reference genome. So we can see that, for example, this sample, a lot of the RNA fragments were not mapped at all to the genome, indicating that this sample is probably not of very high quality in terms of data. Whereas this sample, having more RNA fragments aligned to exonic and intronic regions of the genome, probably has a lot more data that we can work with. As I mentioned previously, our code, rationale, documentation, and other supporting information are being collated in one GitHub repository for publicly sharing and dissemination. And we hope that this will be useful to others who are following PatchSeek protocols and to give them a sense of guidance, a direction for figuring out how to process their data, and as well as getting a sense of the quality control of their data. So this is overall a repo that we are working on right now, and it is in development. So hopefully it is clear that we aim to share the results of our work in an open and accessible manner in accordance with open science principles. Now, there were many lessons or challenges that we kind of faced in working on this project. One of the main examples that I wanted to point out is balancing the comprehensiveness and the conciseness of our documentation. Now, there are many aspects of this, but one particularly important aspect of this that can also kind of inform and help address this challenge is to accurately assess and define our target audience. For example, researchers who have a strong background in coding versus those who do not might require different levels of explanation from our documentation. So it is something that we are evaluating, and it's something that we are taking into consideration in our documentation. So with that, I would like to thank Dr. Shujo Tripathi, the PI of our lab, Dr. Wataru Inoue, who is my co-mentor on my CONP scholarship, as well as the other members of Tripathi Lab and our many collaborators, some of whom are piloting the PatchSeek experiments. And also thank you to CONP for supporting our research, as well as for this opportunity to share our work with me.